Welcome to our video lecture on section 4 of chapter 10 on cancer and the cell cycle. Our learning objectives are to describe how cancer is caused by uncontrolled cell growth, understand how proto-oncogenes are normal cell genes that, when mutated, become oncogenes, and describe how tumor suppressors function. And lastly, explain how mutant tumor suppressors cause cancer. Cancer is essentially the result of uncontrolled cell growth. Normally, our cell cycle has safeguards that we talked about in our last section that are going to help to regulate when the cells divide and when they should stop. But when these safeguards are failing, the cell keeps dividing without control. And so one way this happens is through mutations or permanent changes in the DNA sequence. So during replication, errors sometimes occur, and these could be called substitutions, insertions, or deletions of our nucleotides. Nucleotides, if you remember, are going to be those individual compounds consisting of a nucleoside that's linked to a phosphate. Basically, they're the structural units of nucleic acids, DNA being one of them, RNA being another one. And while most of these are going to be corrected, a small percentage slip through and can be passed on to their daughter cells. So if a mutation alters a gene that codes for a protein important in regulating the cell cycle, it can really lead to faulty proteins. Then these faulty proteins may fail to stop division when they should, or they may overstimulate division, both of which disrupts the balance. So we could see that picture down over here. In our healthy cells, we'll see that there's a balance between our cell division and cell death or apoptosis, and that helps to maintain homeostasis. But if our cell division is going to increase too much, or we have decreased apoptosis, then our cell population is going to expand uncontrollably and we end up with tumor formation. Now let's move on to our proto-oncogenes. Proto-oncogenes are going to be normal genes that code for the positive regulators that we talked about of our cell cycle in the last section. So in other words, they're the ones that are making proteins that help push our cell cycle forward when it needs to divide. And this is important, of course, for growth of the cell, development, and repair. And once again, the problem is going to arrive when a mutation occurs in a proto-oncogene. If that mutation causes the gene to become overactive or expressed at the wrong time, then the gene is going to turn into an oncogene. So this oncogene then is going to act like a gas pedal. And imagine this pedal is stuck to the floor. It's going to keep driving the cell cycle forward, and the cell isn't going to stop replicating when it really should stop dividing. So just to reiterate here, our proto-oncogene is supporting normal cell division, but once it mutates, so we can see that mutation taking place over here, it becomes a oncogene and it's going to trigger uncontrolled cell growth that leads to tumor formation. All right, now that we've looked at our proto-oncogenes as a gas pedal of our cell cycle, let's look at the brakes, also known as our tumor suppressor genes. These produce proteins that act as negative regulators for our cell cycle. And their role is to stop or slow down division when the conditions are not right, or when we see that there's some type of DNA damage that needs repair. One of the most important tumor suppressor genes is P53 that we've discussed before. Again, this is called our guardian of the genome. When P53 detects DNA damage, it can pause our cell cycle and allow it to repair. Or if damage is too severe, we go into apoptosis in order to prevent this faulty cell from dividing. So what we see in this diagram is that if P53 is functioning properly, then DNA damage, okay, or cell cycle abnormalities or hypoxia, meaning we don't have enough oxygen present, will lead to the cell cycle stopping, us repairing that DNA, and then we can restart that cell cycle once the repair is made. Or if we stop the cell and that damage is too severe, then we have it go through apoptosis. But if we have a situation where we have those cases of DNA damage, abnormalities, and hypoxia, and our P53 has mutated or isn't working properly, then the cell cycle continues unchecked, and the damaged DNA is going to be passed on, and that increases the risk of cancer. So mutations in P53 have been identified in over half of all human cancers. So that just really highlights how critical this gene is for controlling cell division.